Okay, folks. We'll start with a sound check. Can you hear me back there? Can you hear me back there? Lovely. Okay. Always let me know if I'm not clear wherever you are. Just a reminder that Susan O'Donnell has been having regular sessions already. Those are Tuesdays and Thursdays at 2:30 at the Heads Conference Room on the Bowen, 4th floor. And Angie Hansen will begin her sessions on a regular basis starting this Thursday at 5:45. So slightly different styles of a presentation, different times. I hope that if you need any kind of help, you'll make sure to take advantage of them or of my own office hours. It seems impossible that it could be this close, but the first exam will be a week from Friday. So I hope you're staying on top of the material we've done so far so that anything new that we cover that you haven't seen beforehand won't take you by surprise. Any questions about DNA structure or any of the other topics before we move into proteins? Okay. Well, let's see if we've done better by this thing. Okay. Close it down. And start this on a slideshow. Come on. There we go. Excellent. That's faster than I expected. And get our pointer going. Okay. So we talked for a couple of days about DNA, nucleic acids in general, about their structure, how that structure provides for the function of storing information, transforming information, how to some extent they can have secondary structures that are different from that. So for instance, like tRNAs. But the nucleic acids are principally informational molecules. They either are actually doing information or they're playing a role in regulating information. There aren't a lot of enzyme nucleic acids, although we're going to see a very important one later on. When you want to look at diversity of function, you have to go to the proteins. Our bodies produce in the order of 30,000 different proteins. And each of those proteins has a particular function, something useful it does for the cell or the organism. And we're going to see that, again, for proteins, as for DNA, their structures are what make possible their functions. That looking at structure will tell us something about function. In the case of proteins, their sequence will then determine their structure. This is in contrast to the nucleic acids, that in the nucleic acid you can have almost any sequence you like and still make double-stranded DNA. By contrast, the specific linear sequence of the polypeptide will have radical implications for the structure of it. We'll be taking a look at myoglobin and hemoglobin as two model proteins. These are very well studied. They keep us alive. That's always a useful property. They're very well known. They are both of them oxygen-binding proteins, but they play slightly different roles in how they deal with oxygen and the fact that hemoglobin can be regulated in its binding in contrast to myoglobin. So let's start meeting some of these proteins and take a little look at it. Here is a space-filling picture of myoglobin. That's one of the ones we'll be talking about in more detail later on. Here's insulin. We don't tend to think of it as a protein, but in fact it is a small protein. You can see the two chains there in this space-filling picture. These are some proteins that we're going to see in more detail as we go along. Chymotrypsin is an enzyme that is going to be one of our models for enzyme actions. Not too big a protein. Here is DNA polymerase, the fragment that does just the polymerase activity, sometimes called Kleinow fragment, and we'll get to know it much better when we do molecular biology later on. Here is maltoporin. This is an example of a transport protein, and we'll talk more about those when we're talking about membranes. Here's a little bit of a collagen molecule. I say a little bit because the top of this molecule hits the ceiling and the bottom is somewhere in the basement. No kidding. And we'll be looking at that more when we talk about structural proteins. Plastocyanin is going to be an important player when we get around to photosynthesis. One of the few proteins that's only in plants, but nonetheless we do study it. Here's phosphoglycerate kinase. This is going to turn out to be important in glycolysis, which is one of the core pathways in metabolism. Yep, let's see. I guess that's it. All right. So, how do you build such a variety of structures? And believe me, I could go on and give you many more. One of the items that is now on ICON under something called Articles of Interest is a poster of proteins that shows you these different shapes in really gorgeous ways. So I will be telling you more about that later. But what we make up these polypeptides out of is a very restricted set of 20 amino acids. We tend to think of those as the only ones that could exist. That's not true. They happen to be the 20 that do get used by our cells for protein making. There are some other amino acids that get used for other purposes. Now, you're going to need to know things like the relative size of amino acids, the relative hydrophobicity of the amino acids, their charge or their polarity. The easiest way to learn those things is by learning their structures. Is learning amino acid structure part of organic chemistry around here? No? No. All right. I'm going to give you a guided tour of the structure of amino acids that'll make it easier to learn it. If you really don't want to learn it, that's up to you. Most people find it's easier just to learn the structures than to try to memorize the properties, because if you know the structures, you know the properties. So let's get to meet these amino acids. There's one cluster that are hydrophobic. So they're mostly carbons and hydrogens, not very polar bonds, and so they don't tend to interact with water very much. And they basically just differ in the size and shape of their side chains. So here's alanine with a little side chain, just a methyl group. Can't get much smaller hydrocarbon than that. Here's valine. Alanine is A, if you use a single letter code. Here's valine. The way they've drawn it here, it's not quite so obvious, but of course, given the bonds of carbon, this is actually a V-shaped side chain. So valine is just the 3-carbon side chain. You have leucine, which is a little longer than valine. This is still the branch, but now it has an extra carbon in there. Single letter code of L. You have isoleucine, which is simply a rearrangement of those atoms. Exact same composition here, just a difference in shape. It's the only difference between those two. And yet there are places where one is crucial and the other won't work. Here's phenylalanine with a phenyl ring off of an alanine. So here, that would be alanine by itself. You put a phenyl ring on, you have phenylalanine. And we're going to call it F for phenylalanine. We're going to see why in a second. Here is one that has a sulfur in it, and its name tells you that. It's methionine. So it's a methyl group on a S-thio atom. So that's methionine, 3 carbons, but an S right, interrupting halfway down. We have tryptophan. This is the bulkiest of the hydrophobic amino acids. It also has a ring. Both phenylalanine and tryptophan have rings, but tryptophan has 2 rings and they're fused together, and there's a nitrogen in the middle of it. And so this is easily the most bulky of the lot. It also hangs off of an alanine, so to speak, structurally. And then you have a very odd hydrophobic amino acid, which is proline. And proline is like a string of carbons that comes back around and bites its own tail on the nitrogen. So it is a ring structure that is a ring back to the fundamental amino acid itself. All the rest of these have their side chain just flapping out in the breeze. Proline uniquely brings its side chain back around to attach onto the amine group of the amino acid. So that's your family. There's only eight of them. They have these relationships among each other that make them fairly easy to learn. The only single letter code that's hard to learn, P, is for proline. That's why we saved P offhand. The hard one is W for tryptophan, and the mnemonic there is twiptophan. I didn't make up the mnemonic. I didn't make up the code. That's what it is. We also have some polar amino acids, and these are all here highlighted in yellow in this case. Here's our second amino acid. It has a sulfur in it. It's called cysteine, so you can still see that kind of thio reference in there. But this one is polar, unlike the methionine. And so if you look here, you see this is an SH group, and that's enough more polar for cysteine to be measurably more polar than methionine. So 2 amino acids with sulfur, one is nonpolar, one is polar. You could have an alanine with an alcohol group. That would be a serine then. Or you could take a serine and add a methyl group. That would be threonine. S for serine, T for threonine. You could have an alanine and hang off of it a phenyl ring and an alcohol. So this is like phenylalanine with an alcohol. This is called tyrosine. It's Y for the Y in the middle because we already used T up for something else. And as you can see, this is kind of an odd polar amino acid because in the back end part of it here, it's not particularly polar. And so you can see tyrosine with this part of it sticking out into the water phase and having that part of it buried sometimes. Here's asparagine, so it's an alanine with an amide group here. You've got the carbonyl bond to the oxygen and an amide here. You can see that that'd be a nicely polar side chain. Oxygen, nitrogen, nicely electronegative. And that's N for asparagine. And we have glutamine, which is simply one carbon longer than asparagine. So these again are partners or twins, if you like, semi-fraternal twins. They're not identical twins. Its mnemonic is Q for cutamine, because G is used for glycine. And glycine is off here in a corner by itself because you can't really call it hydrophobic and you can't really call it polar, and it certainly isn't charged. It's just really, really small. Its side chain is a hydrogen. And as a consequence, glycine is not enantiomorphic. Well, we'll talk about what that means in a second. There's one other of the polar amino acids, and that's histidine. This is, again, a ring structure. Again, this is a heterocycle. It's got two nitrogens tucked into it. So you can see how those would tend to have some polar character to them. Questions so far? Okay.
So you said that glycine isn't polar.
Yeah, it's just really small. I mean, a CH bond isn't going to be particularly polar, but it's too small to be really considered nonpolar. Doesn't repel water at all. And what we're going to find out is that most of these amino acids have places they turn up more often than others. And one of the places that glycine turns up is at bends and kinks. It's a very kinky amino acid. So maybe that'll help you remember it. Not an accident. Now there are some that actually have charges on them, not just merely polar, but are charged. We have two acidic amino acids. So if you take alanine and add a carboxylic acid, we have aspartate, and it's a D for aspartate. Again, I didn't make up these mnemonics. If you add in another carbon in there, you have, again, these kind of fraternal twin relationships of glutamate, and this has the carboxylic acid there. Glu-E, so glutamate is glu-E. And you can see that aspartate and glutamate are cousins to asparagine and glutamine. Asparagine and glutamine are the amide groups. Aspartate and glutamate are the carboxylic acids. Now you know why I was asking you to refresh your memories of all those functional groups. I never say something as a joke. You gotta believe me. I always mean what I say. And then there are two more amino acids that are actually basic. So we've got two acidic ones and two basic ones. Lysine is probably the simplest one to think about first. You have this long hydrocarbon chain, basically, 4-carbon chain, and an ordinary primary amine sticking off the end. And so there's that. And it's K because we already used up L for leucine. So it's the next one down, or one over. And then we have arginine, which is a bit different. It's got 3-carbon chain here, and then this guanidino group, this funny carbon with 3 nitrogens in there. And this is actually kind of an aromatic thing, that it shows the charge being localized there, but it's kind of really the charges on the group as a whole, just like the charges on a carboxylic acid as a whole. And that's them. There really aren't that many. And if you use flashcards, you'll find you can learn a lot of them pretty fast. And then the properties will just carry along with the structures. All but one of them are what we call chiral. That is, the structure of it looked at in one direction is different than if you look at its sister molecule made the opposite way. So this is showing you alanine here. So here's the amino acid part here, and the side chain is the methyl group. If you look at that alpha carbon, that central carbon, there's something different on each of the 4 bonds. And so one way of representing it is that a right hand is not superimposable on the left hand, that it has chirality, it has handedness. Mittens don't. You can wear a mitten on either hand. And glycine is the mitten of the amino acids. Since in glycine, the side chain is just a hydrogen, there's no meaningful way of saying it's left or right-handed. So glycine is kinky, glycine is a mitten, glycine is the odd one out, obviously, in many ways. Glycine's called that because it's sweet. Purified glycine is fairly sweet to the taste. Okay. I talked about the amino acids as being acid or basic as if it was all or nothing, but if you remember from our discussions of pKa and acidic groups and weak acids, for weak acids or weak bases, it depends on exactly what the pH of the solution is. And it turns out that histidine has a pKa of 6. So its pKa is fairly close to the pHs that you might observe in a cell. And what this means is it can be selectively protonated or deprotonated depending on the environment inside the protein and the particular environment within the cell or in the cellular, extracellular fluids. And so what that would look like, here's the base form. It can receive a proton and become protonated. Here it's positively charged. We could consider this as the acid form. It can donate that proton back off and go back to the neutral base form. Since the pK is 6, that means it would be half in this form and half in that form at a pH of 6. So at pH 7, which of these two forms is more common? Are there more protons or fewer protons at pH 7 compared to 6? Fewer. So which form is more likely to be present, the protonated or the deprotonated form? Deprotonated, right. So what you'll find is that at pH 7, roughly 10% or a bit less will be in the protonated form, and most of it will be in the deprotonated form. But the environment of a protein, the actual structure of a protein, can change the pKa of that residue. And though we call it having a pKa of 6 in solution, a histidine group in an enzyme can have pKa's that range from 5 to 8, would you say, Susan? Anywhere from 5 to 8. So the environment of a protein can influence the likelihood of that proton coming on or going off. Yep, there we go. Cysteine is another one that can be protonated in the neighborhood of, physiological pH. So its pK is 8.3. So if you were to look at a cysteine group here, if it becomes deprotonated, you then have a negatively charged sulfur atom there. If it becomes protonated, it goes back to the neutral form. And so as the pH of the solution gets higher and higher, it's more likely to lose its proton. So at 8.3, it's half in this form and half in that form. At pH 7, this will be the form that predominates. So histidine and cysteine are both predominantly neutral at neutral pH, but their pKa's are close enough to neutral to give the cell and give life in general some flexibility in using them as charged groups. Another cute trick that cysteine can do is it can make a bond to another cysteine. So this is a disulfide bond. This is a sulfhydryl group. You could imagine conditions under which you could get those to then make bonds to each other rather than to the hydrogen. And these will turn out to be important as well. I hope you're getting a sense already that these side chains give each of the amino acids a particular character so that they can be used for different purposes. And that's what we're going to see, is that no two amino acids are so similar to each other that there's no difference at all functionally. They all have their role to play in the final shape and function of the proteins. I don't want you to memorize these, okay? But I do want you to take a look at the relative order. We talked a little bit about histidine being close to 6. Both glutamic and aspartic acid have pKa's a good deal lower than that. So those side chains will generally be charged or deprotonated at physiological pH. Cysteine, again, is just to the high side of physiological pH, and then we move on up. Tyrosine actually can be deprotonated, and it depends on the protein environment. It can be better, more so than this would suggest. But the lysine and arginine are the ones that are most dramatic. They have very high pKa's. You'll rarely see these guys without their charge on them. And arginine, look at how much more basic arginine is than lysine. That aromatic guanidino group there gives it almost a full 3 pH units difference. The difference here is that glutamic acid has a slightly longer chain than the aspartic acid, gives you almost half of a pH unit. So the structure of the amino acid will affect its properties. This is just showing this with including here for you the actual chemical structures. The C-terminus of a polypeptide is also an ionizable group. It's a carboxylic acid, and its pKa is actually lower than aspartate. So again, unless there's something else going on, you're generally going to see that in the ionized form, the deprotonated form. We've talked about these other guys. This is showing you what the protonated forms of these are. And so the N-terminus, interestingly, is also, of course, capable of being protonated. Here we go. There's the N-terminus. Got a little bit out of sync there. Its pKa is 9, so it's a good unit less basic than the lysine there. All I've talked about so far are amino acids as individual amino acids. And those are beautiful things, but they don't do much. We won't get structure until we make a polypeptide. And we do that by joining more than one amino acid to another one through a specific structure called the peptide bond. So here's one amino acid, just any old amino acid with an R, another amino acid giving a different R group. And what's going to happen is that between the carboxylic acid of one and the primary amine of the other, we have to remove a water molecule worth of stuff to make a peptide bond. So now you have the carbonyl oxygen bonded to the amide nitrogen, and you have a peptide bond that lies like that. Okay. The peptide bond has a partially planar character to it, and we're going to talk about that in a little bit more. And then the R groups are sort of sticking away from this backbone of polypeptide. This is just showing you how you can have several in a row. And you can see the repeating character of it, that every 3 atoms you get another carbonyl oxygen. Every 3 atoms you have another amide nitrogen. The CD is, I think, especially helpful for looking at this structure. I've alluded to the contributions of resonance briefly before. This is a place where resonance plays a very important role. And so although we draw the peptide bond most usually in this form with an uncharged state like that, this is also a legitimate way of drawing it with the charge distributed differently. So there's now a double bond between the carbon and the nitrogen. And the understanding we have of resonance is that it sort of populates both of these to some extent. And because of that, this bond, the carbon-nitrogen bond of the peptide bond, is a relatively planar bond. That is, it tends to have the carbonyl oxygen and the nitrogen off there oriented in a flat plane. They're not free to rotate around each other because there's a double bond-ish sort of thing connecting them. Okay. So this is roughly planar. This is roughly planar. This is roughly planar. The alpha carbon, the central carbon that holds on to the amino and the carboxylic acid when it's a free amino acid that has the R group growing out of its head. The alpha carbon is not constrained that way, and so that's why they're not showing it in the green. So this is like a sheet that can rotate, a sheet that can rotate. And that one's like a sheet that can rotate, but the alpha carbon in the middle is free to twist all around in terms of its bonds. We're going to see there are other constraints. The only basic limitation to that alpha carbon here, the one that's in the middle, is that the carbonyl oxygens are fairly big. And so they can't rotate in a way that makes those carbonyl oxygens bump into each other much. That's the principal limitation on the alpha carbon, in contrast to the peptide bonds that are more constrained. So we're starting to see already that the polypeptide backbone can't just do any old thing it wants to do. There are certain constraints on it. So we've been setting the stage so far. We haven't actually done anything that's terrifically structural yet except talk about the peptide bond. But now we have the tools to talk about structure. And structure occurs at several levels of organization for polypeptides. The simplest structure is the sequence. The primary structure is simply the sequence of amino acids in a polypeptide. And you can represent it as a linear sequence of amino acids. Here we've got glutamine, serine, phenylalanine, glycine, aspartate. Depending on what that primary structure is, that primary sequence, it may form an area of local order. It may form a region of 5 to 10 amino acids that have a repeating local order right around there. And there are two very common kinds of secondary structure. There are some others as well, but these two are the most common kinds of secondary structure, called the alpha helix and the beta sheet. And we'll talk more about them in a second. Now, depending on how much peptide you've got, what the secondary structure is, and a lot of other stuff, that then can also pack completely up into something that's relatively compact and relatively constant in shape. And that's called the tertiary structure, the packing of the polypeptide in three-dimensional space. The whole polypeptide where everything is when it's all packed up. Now, some proteins flex and bend a bit so they can change that tertiary structure a bit, but it's still going to be where all the atoms are relative to each other in three-dimensional space. Some proteins have more than one polypeptide in them. And if a protein has several polypeptides in it, we can talk then about quaternary structure. If you have two or more, the way that those polypeptides assemble together makes a quaternary structure, the assembly of all pieces at once. So all proteins are going to have primary through tertiary structure. Some proteins will then have quaternary structure if they have more than one polypeptide chain. Okay? Any questions about that before we start talking specifics? Sorry. Okay. So we've talked about primary structure. It's just that sequence of atoms. Secondary structures. We mentioned that the planar carbon-nitrogen bond and the peptide bond limits some of the rotation of the polypeptide backbone. We can easily imagine those side chains, the R groups, might offer limits. I mean, think of how big a tryptophan is or how big an arginine is. That's taking up a lot of space. That'll affect things. But the third thing that affects what kind of secondary structure occurs is the presence of hydrogen bonds between an amide nitrogen and a carbonyl oxygen. So the hydrogen on the amide nitrogen is the donor of a hydrogen bond to the carbonyl oxygen on a different residue. And if you look at this bit of helix here, you can see many, 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 many hydrogen bonds. In fact, every peptide bond can participate in two hydrogen bonds, one up and one down, so to speak, or one to the left and one to the right. And what happens is that the polypeptide chain will take on a minimum strain configuration that in some cases has this repeating order, either of an alpha helix or a beta sheet. We're going to look at that alpha helix in more detail in a second. So these are the hydrogen bonds that are forming in this bit of alpha helix. The alpha helix was the first secondary structure to be described, and it probably accounts for 40% of all protein structure, would you guess, Susan? 40 or 50%. So if you understand the alpha helix, you're well on your way to understanding proteins. The backbone is what is coiled, and let me back up one. You can see here that the backbone is coiling around. It's a helix like a metal screw. It's not a spiral like a wood screw, so it's a helix. It's a right-handed helix, so it's turning to the right as it goes up. There's about 3.6 residues from one complete repeat, and it takes about 5.5 angstroms to do that. The hydrogen bonds run up and down the axis of the helix. They're vertical with respect to the helix. And I'll go back and remind you. So there's about roughly 3.5 residues from here to the next one down. You can see they're not quite staggered. And by having the hydrogen bonds run up and down the backbone, this is what we mean, that they're running parallel to the helix itself, almost parallel. They're running up and down like that. Another feature of it is that the center of that helix is solid. When you show the alpha helix in this ball and stick configuration, it's easy to think that it's, as loose as Christmas tree tinsel, which is very misleading. A space filling diagram like this will make it very clear that the central part of an alpha helix is a solid core. And then the side chains stick out in every direction from it. So as you go around the alpha helix, those side chains are sticking out along the way. The second common form of secondary structure is called the beta sheet or the beta pleated sheet. In this case, the backbone is more extended than it is in the alpha helix. It's as if you pulled it out like this. As a consequence, the repeat unit is different. They can be in either a parallel or an anti-parallel state. So here you can see one particular strand, and the next one next to it is in the same orientation. They both have a carboxylic acid at this end, and their amine groups down there somewhere. It will also work if one is in one direction and the other is in the opposite direction with slightly different spacing. But they can be parallel or anti-parallel. There can be anywhere from 2 to 12 strands in this sheet. So this, if we just had these two, that would be a 2-strand beta sheet. This is a 3-strand sheet. There are structures in which 12 sets of backbones participate in a structure. And in this case, the side chains are sticking out above and below the plane of the sheet. I think the next picture has a space filling. Yes, here we go. So here is the ball and stick diagram of it. You can get a sense of the backbone being more extended. You can see the side chains sticking up and down below the plane of that sheet. And here you can see that the beta pleated sheet, like the alpha helix, it has a solid core to it where the polypeptide backbone is held together by hydrogen bonds. So here the hydrogen bonds are going between the strands in the plane of the sheet, rather than going up and down the helix as they do in the alpha helix. These beta pleated sheets can occur in a number of different ways. So for instance, here we have one that's connected by a simple loop, and that's now anti-parallel because it hasn't had a chance to rearrange yet. If there's a longer loop, you can end up with them in the parallel configuration. There might be just a variety of stuff in between there, and they come back after a long time to be close enough to make a beta pleated sheet. Any questions about either the alpha helix or the beta pleated sheet before we move on? Okay. So alpha helix and beta pleated sheet together account for maybe 80% or more, would you guess, 70% maybe, of protein structure. But by themselves, they don't account for protein structure alone. They have to be put together into an overall three-dimensional shape. And so the tertiary structure will be how you take all these elements of secondary structure and arrange them to make the whole thing. In the tertiary structure of protein, almost all the atoms will in fact have a location. It does sometimes happen that there'll be a tail that's loose and floppy. It sometimes happens there might be 2 or 3 tertiary structures. But it's almost always the case that the atoms have a position. For that reason, it's possible to detect where those atoms are through something called X-ray crystallography. And this is showing you crystals here. Protein crystals can be quite beautiful, and they have a lot of different shape groups. The proteins in this crystal are sufficiently regularly organized that if you shoot a beam of X-rays at it, you can collect the scattered electrons and see where they are. And if you look at this carefully, you can see that some of those dots are very dark and some are very pale. And through interesting mathematics, you can take that pattern of dots and reconstruct where things had to have been to scatter the electrons that way. And the analogy you can think of is this. Have you ever been driving by an orchard and you look down the rows and you can see down the rows at one angle, and then you keep moving down the road and now you can see down at a different angle? That's a little bit of an analogy to what's happening with the X-rays, that the way the trees are planted in the orchard affect the way you can look down the rows. And the packing of the atoms in the protein affects where the X-rays can go. Protein tertiary structure is grossly separated into what's called globular proteins, which tend to be globular, and fibrous proteins that tend to be long and skinny. And we'll be looking at some fibrous proteins in a little bit. When we look at protein structure through X-ray crystallography, we can resolve that position in greater and greater detail. And the more detail in which it's resolved, the more thoroughly we can view the actual details of that tertiary structure. So here it's been resolved to 5 angstroms, and you have a broad sense of where stuff is, but it's fairly messy. With a better crystal, with more powerful X-rays, with better detectors, you can then increase the resolution of your image. Here it is down to 3 angstroms, and now we're getting down to sort of see not merely the general backbone pattern, but where some of the side chains are. And then if you continue your resolution, now you're basically identifying the location of pretty much all the atoms. In the DNA slideshow I had last time, there is a picture of DNA crystals that shows where the water atoms are in the minor groove. And so they are sufficiently steady in their positions to be able to identify the water molecules. In broad terms, we can say that tertiary structures usually have a hydrophobic core and a hydrophilic surface. So of all the side chains, you can sort them out into thinking of them as more hydrophobic or more hydrophilic. The more hydrophobic ones will usually tend to be here in the interior, and the hydrophilic ones on the exterior. I believe in this picture the blue has been the code for the hydrophobic, and the red is for the hydrophilic, and the grays are ones that are not particularly one or the other. Some proteins, in addition to having a polypeptide backbone, will have a prosthetic group. And here you can see the prosthetic group in myoglobin. Heme comes from the word for blood, and hemoglobin is the globular protein from blood, and the heme group was first isolated from hemoglobin. We're going to see the heme rings in many, many different locations. And when we look at its structure in more detail, we're going to see that the structure of the heme ring allows it to do chemistry that the side chains can't. So although we have these 20 amino acids in their side chains, and they can do a remarkable variety of things by themselves, if you add in prosthetic groups like heme, like vitamins, like cofactors, like metal ions, the range of the chemistry goes up dramatically. And so the prosthetic groups are helping the protein to do something, like a prosthesis can help someone walk or whatever. This gives you a little bit of a sense of different ways to represent a protein. All 3 of these are myoglobin. Here's one that's just showing you the wire background, if you like, which shows you the exact kinks along the way, but doesn't really bring out the helices too well in a flat picture. This is sometimes called a ribbon diagram. It's kind of a convention, but it lets you see that helical structure more easily, especially if you're just looking at it flat. And here in the space-filling diagram, you noticed all that blue stuff there. I was talking about hydrophobic stuff being in the interior. Why is there so much blue stuff at the surface? Well, here is the heme group, and we're going to talk about the hydrophobic pocket that the heme group has to fit into and why it's there. So all of these features are telling us something, but different aspects about it. Any questions about that before we move on? Okay. Let me just see something. That's here. We've talked so far about the idea of compact structure and so forth, and it turns out that you can sort of subdivide the idea of tertiary structure into what are called domains. The tertiary structure will be the whole packing of everything of a single protein, but when we look at particular proteins, we can often find regions of that tertiary structure that fold as a unit by themselves, so to speak. And so in this particular example that's given, they're showing you a green domain and a red domain. Sorry about that if you're red-green colorblind. But you can see that this domain is only connected through this little flexible tail there, and then this domain is there. This whole thing is likely to be the functional protein, the functional tertiary structure for this protein. And yet, in many cases, if we come in with a protease, we can nick that little tether and now have the two domains separate from each other. And in some cases, those domains retain interesting properties. So for instance, if this were a DNA activating protein, part of this might bind to DNA and the other part might activate some chromosomal protein. And in that case, it might well be possible to split it there and still bind to DNA with no activation, and then activate it if we have some way to tether it down or tie it down. Here's just another example of a two-domain protein. You notice how there tends to be some sort of surface area here. It's not at all uncommon for there to be complementary surfaces for the domains to interact at. And we also have good reason to think that this is one of the features of evolution. That is, we find domains of proteins being reused in different settings. So once one particular domain comes up with a cool solution to a particular problem, you may well then see it used in other contexts for a related problem, or sometimes for a surprising adaptation. Any questions about tertiary structure in general that we've talked about so far? Let's talk a little bit about hydrophobicity. I talked, when we were talking about the amino acids, about there being hydrophobic and hydrophilic amino acids, and that's a good first approximation of just saying one or the other, or hydrophobic and polar. But nonetheless, you can imagine that the particular structures of it make a difference of degree, and that's what a hydrophobic scale is trying to capture. It's asking the question, how likely is it for this residue to be in water or to sort out of water? And the scales that people have come up with are generated in a number of different ways and can give slightly different or sometimes interestingly different answers. This is a scale that's a little different than the one in your book. This is looking at the hydrophobicity of it as a residue in a polypeptide, rather than as an amino acid. And the two scales are comparing either what's called an interface, the tendency to sort out into water or out of water at a water hydrophobic interface, like the vinegar oil interface in salad dressing, versus the tendency to sort into octanol, which is a straight solvent. And if we take a closer look, you can see that the most hydrophobic of the amino acids are the three that are aromatic hydrophobic amino acids. So you have tryptophan, which is the most hydrophobic no matter how you measure it, and that makes sense. You've got those two rings you've got to cope with. And then you have phenylalanine and tyrosine as the next two. Interestingly, if you're trying to force it into octanol, you see more of a difference now between phenylalanine and tyrosine, because tyrosine has that alcohol group that's got to be accommodated, whereas in the interface scale, it can just stick its toe into the water and not show up that much beyond that. The next four are not nearly as hydrophobic as the three aromatics. And so it's not nearly as surprising to find them either near a surface or at a surface. They'll tend to sort away, but they're not going to be vehement about it. And you can see here methionine, cysteine, isoleucine, and leucine. Interestingly, down here with the octanol scale, the cysteine, like the tyrosine, is showing less hydrophobicity because the cysteine has that sulfhydryl group that it can stick its toe into water up here, but it has to cope with it on the octanol scale. If we go at the other end, at the charged end, it's not at all surprising that the charged amino acids are going to be very hydrophilic. You've got a whole charge you've got to accommodate, and nonpolar things, don't deal with that at all well. So you can see that aspartate and glutamate are the two most hydrophilic. Very, very uncommon to find those buried unless there's a real good reason. So if you see either of those guys buried in the interior, you can go, oh, I wonder what it's doing there. It'll almost always be something interesting. The two basic ones, again, are much more hydrophilic than the sort of intermediate range of stuff. But again, when we try to force it into a solvent, these things are more or less hydrophobic, if you like, than when you give it an interface, because at the interface it can sort between the two of them. So this should give you a good sense that there are strongly hydrophobic amino acids, strongly hydrophilic amino acids, and then ones in between that can certainly be seen as more or less. I mean, clearly leucine is going to be more hydrophobic than, glutamine, but they're not so radically different from each other as the aromatic hydrophobic ones and the charged hydrophilic ones. You can actually, here's proline showing up as a hydrophilic, or just shy of it, I guess. And it can be found at the surface of proteins, but it can also be found in the interior if you have to make a funny corner. So here are, in myoglobin, what they're trying to highlight here are the hydrophobic and the hydrophilic residues. And here you can see the hydrophilic residues in a nice bright angry red. And here you can see hydrophobic residues in a sort of more reticent retiring green. But there are some residues, if you look carefully, that are neither red nor green. So here's one that's gray here, and it's gray over there. Here's one that's gray here, and it's still gray over there. I think, let's see, over here, no, not that one. I think that one maybe, yeah, that's another one that's neither one nor the other. And so you can see from those hydrophobicity scales which amino acids those might end up being. Okay. Let's call that quits for the day, and we'll start talking about protein folding on Wednesday. Do remember that we have office hours coming up Susan will be meeting on Tuesday. Oops. Susan will be meeting Tuesdays and Thursdays, and Angie starts on Thursday.
Good, yeah. Keeps me thinking. Okay.